Hello and welcome everyone. We are glad to come back online with our series of lecture, and that is Pharma Expert Talk. I hope everyone can hear me, right? Just comment it over here. So I will just make sure and go ahead with the session ahead. Okay, great. So there is a worse situation of COVID-19 outside. I hope you people are being safe and taking care of your loved ones. And we have got somewhere around more than 2,500 participants from various industrial fields, academics, and definitely students. And I welcome all of them. And believe me, we are going to learn important aspects of research activity, that is technology transfer. Before going ahead in the sessions, I'm loving to inform you all that we at Pharma Tutor are organizing series of lectures from various industrial experts who guide students and other pharmacy professionals about career prospects in various fields of pharmaceutical industry. So far, we have invited talks from many reputed industry professionals, and they guided on career in clinical research, pharmacovigilance, production, QA, IPR, regulatory affairs, etc. So if you have missed those lectures, you can definitely watch out those videos available in our YouTube channel, and surely you can subscribe and click on bell icon so you will not miss any such videos in future. Okay. Students who are from B Farm may not aware about much about technology transfer, but I'm sure that M Farm professionals are very well versed with this term. But believe me, you are going to learn more about in today's sessions. And as you people are aware that pharmacy is field of innovation and research, and we all are aware about that there are separate research labs are there with many big pharmaceutical industries, and they have in-house tech transfer team also where pharmacy professionals are employed. Also, a bit as today's competitive scenarios, many pharmaceutical industries many pharmaceutical institutes are also indulge in research activity, and they also require technology transfer for commercialization of their product. So in brief, if we want to say, technology transfer is not only deals with IP protection, but it also deals with efficiency of process, it maintains the quality of product, and it provides cost effective production. So with this brief talk, I'm just going to invite our first guest for today's session, Dr. Sanjay Bharadwaj sir. Hello sir, good morning.
Yeah, good
Good morning
morning.
sir.
Good morning.
Good
Good
morning, good
morning.
morning. Thank you for coming at Pharma Expert Talk, sir. Before just we are handling to Sanjay, sir, I just want to introduce you people about Sanjay, sir. That it's my great honor to introduce him. Actually, he's obtained his PhD from IIT Mumbai. He has received Emerald Outstanding Doctoral Research Award sponsored by International Journal of Operation and Production Management, which is a top rank official journal of European Operation Management Associations. And currently, sir is leading a technology transfer as scientist F and IP management related activities at ARCI, which is the International Advanced Research Center for Powder Metallurgy and New Materials at Hyderabad. He is the chairman of Indian Institute of Chemical Engineers, Hyderabad. He has around 24 years of rich experience in domains of technology transfer, collaboration, IP valuation, monetization, knowledge and management of nanoscience technology, production planning. Um, he has involved in forging of more than 190 collaborations or technology transfers for ARCI with startups or some established companies, academic institutions, other R&D centers, and there is a lot more to talk about, sir. But definitely, in brief, I'm concluding that you people are going to witness one of the, we can say, topmost professional in technology transfer, and his topic of discussion is technology transfer and commercialization. With this, sir, brief introduction, I just love to hear from you, sir.
Yeah, surely. Thank you very much, Rajesh ji, for nice introduction. Then I will share my PPT.
Yes, sir, it is available, sir.
It's fully on the screen.
Yes, sir, it is there.
Okay. Good morning, all. Good morning, everyone. And I am thankful to Pharma Tutor for inviting me for this very interesting topic for all of us who are working in the field of innovation and technology transfer and commercializing and monetizing this research. So I am glad that the response was huge. I was told that almost nearly 2,500 uh, participants from academia, from industry, and then from in R&D labs and different stakeholders. They are attending this talk. So I welcome all of you. And then yeah, it's a big honor to talk to you, to interact with you, including very esteemed guests, very honorable professionals in the field of pharma. So to start with,
Yes, sir, continue, sir.
slides, just a minute. Slides are. Is it visible now? Next slide.
No, sir.
Is it
Yeah,
visible now?
yes. Now it is visible, sir.
Okay. My structure of presentation is like I'll talk about ARCI. This is the lab I work with. Then IP development in diagnosis-based collaborative and technology transfer models, which we have been using from last more than 6 years to bring in efficiency and effectiveness in the process of commercializing IP we generate in the lab. Partnership trends at ARCI, we have compared whatever efforts we have put in during last 6 years. What is the outcome of these efforts? That I will discuss in brief, and then some case studies from healthcare sector, some pharma kind of sectors. I'll discuss with you, and then some concluding thoughts about ARCI. ARCI is an autonomous R&D institute of Department of Science and Technology, Government of India. We develop, demonstrate and transfer technologies in the area of advanced materials and processes. We provide technological solutions to industry and strategic sectors. We provide training, high quality training in the area of advanced materials and processes. Our research activities are in major four verticals. One of them is nanotechnology, in which we have research in the areas of nanomaterials and carbon materials. In case of coatings, we have again three sub areas: engineered coatings, laser processing of materials, and sol gel coatings. In case of ceramics, we have oxide and non-oxide ceramics. In case of energy, we have fuel cell, solar, and automotive energy materials. What we do, we transfer technology. This is the topmost priority for our lab. Then we do undertake collaborative research with different organizations who have complementary skills and capabilities. Then we have sponsored research, which is generally funded by government agencies, different government departments. Contract R&D is taken up at the instance of companies both from India and abroad. Then to accomplish all these tasks, which are on the left side, technology transfer, collaborative R&D, sponsored R&D, and contract R&D, we have very nice facilities for upscaling of technologies, to have a limited production which can be used to test the products at pilot scale. We provide consultancy in the areas of competence, whatever I just now mentioned in the last slide. And then we have very nice facility to do search for patent and literature, and then to test and characterize those materials. We have one of the best facilities in the area of materials. Till now, ARCI has entered into 40 agreements for development, demonstration, and transfer of technologies. We have provided more than 250 technological solutions to industry and strategic sectors. We have trained more than 2,000 students for their PhD, master's, and bachelor's degree programs. We have field tested technologies in multiple sectors, right from aerospace to textiles. They help in two different ways. They help us to de-risk the technologies, and they help us to make a good business case, which will be convincing a company to invest for further upscaling and setting up a manufacturing plant. We have two kinds of programs in the lab. One is in-house, other is on-demand. In-house is something we initiate on our own, keeping in view our country's industry requirement in mind. Maybe 5 years from now, what will be required by the industry, something of that type. And then on demand is something when an industry comes to us and then they ask us to start something. This is on demand programs. We believe in collaborative approach in our upscaling as well as technology transfer. That's why we have forged partnerships with academic institutions, R&D labs, and industry from India and abroad in over 15 different countries. Then next is I will talk about IPDI, in IP development indices based research collaboration and technology transfer models. Before discussing the main model, I would like to sensitize you on few related issues. Can we think of research upscaling without understanding the nuances of a related domain or sector or a field? That's not possible. We have to have understanding of a particular domain in which we are planning the upscaling of research before we think of making a model. The next is we have to consider the mandate of the organization, what role this organization will play in the upscaling process. For example, industry will play a different role, a research lab will play a different role, an academic institution will play a different role. And we have to understand that mandate before we envisage the role of that particular stakeholder in upscaling process. The other thing is whether diagnosis is enough. Diagnosis is something like we assess the readiness level of a research program. That's not enough. We should also think of some treatment, because you are today all our friends are from pharmacy background. They can understand this much better than my colleagues in other engineering areas. Just assessing a readiness level is like a diagnosis. And that's not enough. We have to find some support systems, some complementary capabilities, so that this program can be made more useful. Can we treat all partnerships in the same manner? For example, can we treat all problems, all diseases in the same way? Not possible. That's why partnerships, whenever a research lab is partnering with the research lab is partnering with an industry, research lab is partnering with academia, they all have to be treated in different way. We have to envisage different partnership situations, and then we have to provide models so that all the partners who are coming forward to do some joint work, they feel happy that they can reap the fruits in a very fair and just way. Then we have to apply different thought processes for forging partnerships and identifying value addition needs at different readiness level. When you are having a research at early stage versus you have research at pilot scale, or maybe your research is then ready for transfer to industry, at all different stages, your thought process has to be different. Now let's see some examples. Now how things are emerging. If you see electric mobility and solar, we find multiple areas merging. When we find innovations, they are being practiced in this area. If you see now AI, ML, 3D printing and high strength materials, they're merging again to impact the manufacturing sectors. Enhanced realities coupling with the education imparting tools. AI, robotics and sensors, they're combining with medical technologies for giving better treatment in neurological, heart and complex surgeries. That's how we see. And then drones, which is probably you can appreciate, drones are being used now for delivery of medicines, food, and other essentials in difficult to reach areas, which are already being used in photography and farming. If you see all examples, there is kind of merging of emerging technologies. Different emerging technologies are being merged, converged to make products which can be used by society, which can be used by our nation to make life of people better. Now, other aspect, can we treat all enterprises in the same way? For example, traditional small and medium enterprises, they are flexible, but they have low absorptive capacity for innovations. Their understanding how to manage relationship with the research labs, how to manage relationship with the academia. Still, there's a need to do some work in that direction. Technology based startups, they're flexible, they have good absorptive capacity because they're mostly led by technically qualified people, but their financial capacity requires some attention. Large companies, they deal with rigid structures because they have legacy of the old complementary assets, big bureaucratic uh, structure. Then how they're handling this kind of handicap of bringing innovations in their companies, large companies, their absorptive capacity, they're improving by having the research facilities in the research park or incubators of, for example, in IIT Madras we have research park, we have large companies setting up now the research wing in those park, and then those teams are encouraged to have joint research programs with the faculty members of IITs. Similar thing is happening in IIT Bombay, IIT Kharagpur, and other IITs. Now, coming to this model, we see here we have divided at ARCI total process from ideation to technology transfer and commercialization in 10 different steps. The step 1 we call IPDI 1, IP Development Index 1, is associated with basic concepts and understanding of underlying scientific principles. What you do here is you study the related literature and patents and then find possible applications of whatever research you are envisaging. And then just to briefly tell you, at level 1, many IP firms, IP companies, patent related companies, they do analysis called white space analysis. Heat maps they make to identify the spots in which there is a possibility of doing research and that there is a possibility of filing patent. When you reach level 3, you do a technical feasibility analysis and then link that with the targeted applications. Level 4, with a coupon, you do analysis. Technical feasibility is something like whatever parameters you identified in step 1 and 2, you see in the lab whether those parameters are affecting the process in the way in which you are thinking. Level 4, you do coupon level testing. Coupon means it's a miniaturized version of real life component. And then you simulate the conditions in which you are trying to develop a component in the lab, or maybe in case of pharma, it can be some drug. You simulate the conditions in which it will be tested in the lab itself. And then level 5, you repeat. It's like robustness at the coupon level, small level. Level 6, you do field testing with a real life component or maybe real life drug. In case of pharma, we can call it clinical trials. And level 7, you repeat those clinical trials, or maybe in our case of materials, when they go to different applications like aerospace or automotive, you repeat or you check the consistency in the field conditions by trying out different components. Similarly, like clinical trial, we do multiple phase 1, phase 2, phase 3, and then we do in animal, we do in human being, and then we say, yeah, now it's safe enough to be administered to a human being. After this, we make a business case. We assess the potential techno-commercial feasibility. We see from the angle of intellectual property, IP, whether or not your innovation is infringing some competitor's patent, and then you will have a strategy to how do we surmount those kind of challenges. Then safety angle analysis, then from angle of health, then from angle of environment. And then after you are convinced of the feasibility from all possible angles, you initiate technology transfer. And then at level 10, you support a company which is taking your technology in stabilizing production. Then level 1 and 2 form the part of exploratory studies. Level 3, 4 and 5 form part of laboratory experimentation. Level 6, 7, 8, the form of part of field trials. In case of pharma, it can be called clinical trials. And level 9 and 10, when your research unit is transferring the research result to a unit which is making manufacturing, which is making some product, then it's called technology transfer. Then we find is the lab industry interaction and the very close partnership is very crucial to cross level 6, 7, 8, 9, 10. This is, we have named, actually we've written some papers in this area, we have named these stages as technology valley of death. Now if you see, other point here is, this model combines both outside in perspective and inside out perspective. Outside in means what is happening elsewhere, what's happening outside in the marketplace. That kind of thing, when you implement this model to do research and then which is aimed at doing technology transfer, you have to understand what's happening outside, and then you will reconfigure or maybe change the programs happening in the lab so that you can tune yourself with whatever is happening outside. Then you couple that with the inside out perspective, because again you should understand what's happening in the lab. Without understanding your lab's research, you can't advise, you can't give them suggestion how they should change, how they should fine tune so that it can be made useful. So then this model helps you to combine both outside in approach. You have to understand the outside marketplace, outside environment, as well as inside lab, what's happening, both understanding of both systems and thoroughness in that will help you to have effective research, collaborations, and technology transfer. Now, let's understand this aspect, diverse expectations of industry, R&D, and academia. If we see R&D readiness, industry wants exact information, how much time it will take, what readiness level you are. In case of applied R&D, now tools are being developed to assess R&D readiness. In academia, again, they are also developing tools to assess readiness of research programs. Roadmap from starting the partnership to envisaged goal. Industry wants very clear roadmap with timelines, very clear timelines, estimated timelines and the roles. But then for a research lab and academia, it's difficult because uncertainties are involved in the research, whether it's early stage, even pilot scale also, so many uncertainties are involved. Time till realization of optimized process and saleable product. Let's see on this parameter. Industry wants product as soon as possible. They want to optimize the process as soon as possible. But when it comes to research lab, you just think over, they will find difficult to commit because so many uncertainties are associated with IPDI. Whatever I discussed with you, IPDI 5 means lab scale results are repeated again and again. Level 6, 7, 8 means pilot scale, your field trial, your clinical trials. You repeat your clinical trial, you make a business case. All that has so many uncertainties. And that's why in a research lab or academia will find it difficult to commit. The lead responsibility in the IP value chain, from IPDI 8 onwards, IPDI 8, just for your recall, it deals with the making of a business case. The lead responsibility beyond IPDI 8 is being taken up by industry, industrial organization. Applied R&D will lead the program from level 1 to 7. One is like ideation, and 7 is you repeat your field trial. In case of pharma, repeating the clinical trials. In case of academia, generally they will have interest in IPDI 1 to 5. One is ideation. Again, level 5 is you have lab scale experimentation, repeating them in the lab. Major driving force for industry's monetization. They will not value generally till something will give them in the end the financial results, financial returns to the company. In case of applied R&D, the parameters, they are looking for patents, they are looking for publications, but they are looking for equally uh, well this monetization also. In case of academia, generally they were looking till now at publications and patenting, but now gradually they are also looking at monetizing the research happening in those academic institutions. Flexibility to engage with the IP value chain. Industry has high willingness to leverage R&D labs IP, but they depend on the frameworks. So we as research labs, we have to create the frameworks which are giving flexibility to those industry partners, which will give them the hope of giving the results in a fair and just way. And then the same way, academia also has to create the frameworks and models and the templates for different partnership situations, which will enthuse industry to forge partnership with the academia or R&D. Now, why we need these models? I have been emphasizing that we require IPDI based collaborative and technology transfer models, because research results of public funded R&D is a philosophy. Public funded R&D should benefit public or society or nation. And without having right models by which you can partner with the possible beneficiaries, how do we show the results of the research? That's why there's a need of having partnership models which can address different possible situations of collaborations and technology transfer. What should these models do? They should provide enabling frameworks and then agreements for win-win partnerships. They should help us to forge partnerships at all levels, from ideation to pilot scale to the technology transfer. They should provide us the wider choice of potential collaborators, and they should help us to converge the diverse motivations of stakeholders. I have mentioned earlier that the stakeholders like industry, academia, R&D, they have diverse motivations. These models, which bring them together, they should capture the diverse motivations of these stakeholders. Now, considerations in choosing appropriate models. Readiness level. While we initiate partnership, what is the target readiness level? We should keep in mind these two things: at what level we are forging partnership and at what level we want to reach. We should keep in mind the activities of partnership stakeholders. For example, R&D lab versus academia, they will have different models. R&D lab versus industry will have different model. We should have that kind of mandate in mind before we propose a model to have a joint activity with these stakeholders. And then imprecise assessment of R&D level will pose difficulties in choosing an appropriate partnership model. We should have mechanism to have precise assessment of R&D level at which we are now, and how we should make a roadmap also. We should be clear. And then if we have imprecise assessment of R&D level, then we will make a wrong roadmap, and then it will lead to possibility of confrontation or maybe some kind of disharmony is higher if we don't have a precise assessment of R&D level in the lab when we are forging partnership with the industry. And then we should have sector specific nuances in mind. For example, pharma, advanced material technologies. For example, in our case, we should keep what kind of, how they upscale, how the technologies are upscaled in these sectors. Benefits: if we use these models, we can optimize the utilization of available IP in the labs. We can identify the validation requirements, support systems, and complementary capabilities. We can make a clear roadmap of financial compensation based on the milestone linked deliverables. We can have transparency in negotiations to improve mutual trust. And then we can have flexibility to engage different stakeholders from industry, academia, R&D, if you use these models. Now, usual TRL scale is this actually, which is given in the websites. If we compare this TRL scale and the IPDI based collaborative and technology transfer models, we find that demarcation between levels is here, it's not so precise. It's slightly if you compare with the IPDI based collaborative and technology transfer model, it is partial. When it comes to identifying partnership needs, this model, TRL model, it doesn't really discuss about how do you forge partnership at different levels of readiness. Then capturing nuances of pharma and advanced material field, or maybe other field for that matter, yeah, it doesn't capture those nuances. And guiding a lab having technology transfer as a mandate, it has partial inherent features in guiding such lab which has a mandate of technology transfer. Now coming to this model again, we have seen that 1, 2, 3, 4, 5, they form exploratory studies and laboratory experimentation. What kind of questions we should ask here is, what are the avenues of research, whether your work can be patented, what kind of competition exists, whom I may collaborate with, what kind of partnership models exist, and the cost and valuation of IP. Then at this stage, a research team has to answer these questions, and then that's where I think role of people who are in IP groups or IP teams or patent firms, they can assist research groups, because they will not have wherewithal, they will probably will not have time to do this kind of analysis where we can get answers of these questions. For example, avenues of research, we have to do white space analysis or heat maps. That's where the role of IP groups, either in-house or IP groups or maybe outside IP firms, whether their work can be patented. Yeah, again, we have to get answer of this. Competition, we have to study the market and then to get idea of what kind of competition exists, and then who are possible partners who have complementary skills and capabilities whom we can partner with. That kind of answer we will get from competition analysis. And then after understanding of this total landscape, we can think of having partnership model which will help, which will facilitate the partnerships in different situations. And then cost and valuation of IP, and then cost estimates of technology derived products, a road mapping. By the way, you're forging partnership at this some level, at level 3, 4, 5, then you have to make a roadmap to the level at which your partner is interested to work with you. And then scenario building. So then these are the different models we have developed at ARCI. Just to tell you briefly, cooperative R&D model we use when industry is a partner. Then consortium model we used when we have different partners from academia, industry, and R&D. Interinstitutional model we are using when we have other R&D lab or other academic institution as a partner. And then sponsored R&D model we are using when a government agency is sponsoring research, and then contract R&D is being used when an industry partner is sponsoring or maybe funding research which is maybe at level 1, 2, 3, 4, 5. Level 6, 7, 8, they constitute field testing, as I mentioned earlier. At this stage, the inputs or questions which we should pose to our research teams, and then we maybe IP groups and research teams can jointly find possible answers of these questions. They are, whether a product can be launched, what are the patenting possibilities, because you have to continuously strengthen your patent portfolio. What about the possible partners for validations and effecting technology transfers? Different models to forge partnerships, and the cost and valuation of IP. At this stage, you have different collaborative models like demonstration and transfer model, which is used when an industry partner comes to you then as a lab, and then you have to demonstrate and transfer technology to this industry partner. Joint demonstration is done when a company or maybe some university has already done work till level 5, means they have done lab scale work. They want to jointly work with, maybe in case of ARCI, our lab, and then we jointly do 6, 7, 8 level of work, and then we take a call how to transfer this technology after this work of 6, 7, 8. It's like field trial, or in your case, in pharma case, it's like clinical trial. Knowledge transfer, sometimes some companies are looking just for knowledge transfer. They want a knowledge document of whatever work has been done till level 7, and they want to use this for their own objectives. Option model we have used maybe last few years to encourage industry, give them a lot of flexibility. We give them 6 months time to 1 year time to decide whether they want to go ahead for technology transfer or vice versa. They can take a product based on the technology of ARCI. They can field test. After the feedback from the marketplace, they can decide whether some kind of fine tuning is required. They'll give feedback to the research teams, and then teams will fine tune. And then based on the outcome of this field testing, sensitization in the marketplace, this partner company will decide about the future course of action. Application development and product supply for niche uses. Sometimes a partner industry or partner organization, they are just interested in application development and supply of some products for niche uses. At level 9 and 10, we initiate technology transfer, and level 10, we support this partner in stabilizing production. At this stage, there is a need of product launch, and then analysis with respect to what kind of markets they should tap, this technology and associated product should tap. IP analysis, whether infringement is being done, then how do we solve this kind of challenges we should do. Freedom to operate analysis, whether you can operate or you can commercialize the technology without infringing the patent of competitors. Patenting possibilities, and then possible collaborators, technology receivers, and then partnership models, and then cost and valuation of IP. At this stage also, sometimes what happens, option and technology transfer both can be used, both the models can be used. Sometimes what happens, although we have done work till level 8 to make a business case, but then industry partner may like to have an option model to be used. They would like to test again in the markets or the applications which they have in mind, and then they will make their own business case, and then they will decide about technology transfer. Now I have attempted for pharma sector how we can like capture different activities at different levels. If you see at level 1, 2, 3, 4, 5, they capture both exploratory studies and lab scale experimentation. Here, in case of pharma, we aim for pharmaceutical development. Then we develop a pilot process, keeping in view the intended performance of the drug. We design a quality product. We adopt quality by design. We define design space, wherein we define critical process parameters and critical quality attributes, keeping in view the regulatory assessment and approval. And then we have to keep in mind the ICH guidelines also. Coming to level 6, 7, 8, we find we have to do formulation development, process development, and developing of techniques for analysis. Technology recipient, if we identify at this stage, that will be useful because they will provide useful inputs for further effectiveness in the transfer. Identify control variables like CPPs and CQAs, and then sources of variability by process monitoring, and develop a control strategy by process analytical technology and pie data acquisition kind of techniques are now available to have a good implement a good control strategy, and then build, develop, and update a transfer master plan. Then coming to now level 9 and 10, if we see the pharma sector, we have different situations when a research lab may be transferring a technology from R&D to industry. It can be in-house R&D or maybe external R&D. Then academia to industry or industry to industry. Then your transfer may include product and process. For the analytical method, it can be same or equivalent, and engineering. And a team has to be formed of multiple discipline professionals like process, quality assurance, regulatory, and operations. And it should be product specific. And then this team should have meetings frequently so that they can iron out the possible difficulties being faced in the upscaling process. Then demonstrating consistency and reliability by repeat batches, by making repeat batches. And then manufacturing validation batches to comply with the regulatory submissions. And then at this stage, we should also have facilities for manufacturing which will comply with GMP requirements, good manufacturing practices requirement. And then 5W are very important at this stage: what we are making, when we are making, and why we are making, and then where we will make that. Because in case of pharma, it's very important what kind of facilities are required to manufacture and by whom it will be made, because the talent required and by the kind of human resource what you will use, that's very important. And then 1H is also important because know-how is extremely important. Whatever know-how is being developed by either in-house R&D or by external research lab, that is also extremely important. That kind of understanding has to be there before we transfer the technology for manufacturing. Now, this ICH guidelines, International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use guidelines, ICH guidelines also prescribe that technology transfer can happen in different situations. It can happen between R&D and manufacturing. It can happen between different manufacturing sites to realize products. And then it has pharmaceutical quality systems, control strategy, and then manufacturing. Then we have used, as I mentioned earlier, that we have used this model almost nearly 6 years ago. Then we have compared the critical parameters between last 5 years, means 10 to 15 versus 15 to 20. We found that there is nearly 2x growth. Our collaborations and technology transfers have become double in period of 2015 to 20 versus 10 to 15. And the companies who have signed our confidentiality agreement with ARCI, they also became double, who are exploring possible partnerships. If you see ARCI approach, we have of our ongoing research programs. Then we couple our research program with the end users for field test, and then possible entities who can commercialize the research. We try to couple with both, and we have to make a distinction between the two. Actually, for example, any component can go to maybe hospitals. In your case, clinical trial, some patients, or maybe hospital, but they will not manufacture. The product will be manufactured by some drug company. So we have to make a distinction between the entities who will do field test versus the entities who will commercialize the research. And in this kind of couplings, your ecosystem influencers will be very useful. There are different government programs who will help us to forge partnerships, and this will lead to products and services for society. And then if you see the products and services for society will lead to wealth creation, and wealth will be used to again fund the research programs to develop new technologies, and this completes the circle. So if your circle is having this kind of weakness, it's not complete, then it will create a deformity in this total loop. Now, case studies. I have 3 case studies to discuss with you today. One is on disposable blood bag system. Other is on liposomal M4 tericin B, and then UVC-based disinfection trolley. Disposable biomedical plastic bags are used for collection, storage, transportation, and transfusion of blood and blood components. They don't require exhaustive cleaning, rinsing, and autoclaving and breakage problems. An institute called Shiraj Chitra Trinal Institute of Medical Sciences and Technology, Trivandrum, they developed this technology at the expenditure of 0.2 million, funded by NRDC, which is organization again part of Government of India during 1977-78. And then this institute assigned this technology to NRDC for transfer of technology. 1983, the first technology transfer happened to a company called Peninsula Polymers, Trivandrum. Now it is called Terumo Penpol Private Limited. This company has 25% equity by NRDC and KSIDC. Technology transfer involved 0.3 million rupees as fee and 3% royalty for 10 years. And it was transferred on non-exclusive basis. Non-exclusive means it can be transferred to many companies. In 1991, the company called Hindustan Latex Limited was the second technology receiver, and then they paid 1.65 million rupees as know-how fee, and then 3% royalty for 10 years, again on non-exclusive basis. In 1994, two other companies received this technology. One is J-Mitra and other is Electromedical Industries. Each of them paid 4.5 million rupees as fee and 3% royalty for 10 years for non-exclusive transfer. Then in this case, 1987-88, production was stabilized and was conforming to GMP requirements. Product was launched in the market at rupees 10 per bag, vis-a-vis rupees 30 per bag from Japanese competitors. And what happened after Japanese competitors saw that Indian companies supplying this product at rupees 10 per bag, they started supplying or dumping in the Indian market at rupees 10, rupees 8. Then NRDC took this case to government and then convinced the government to impose anti-dumping duty. And then after this anti-dumping duty, this product could be marketed by this company. And then now it's being exported to various countries like Africa, Southeast Asia, and Europe. Other cases about liposomal amphotericin B. This is antifungal agent. It is having limited applications. It was having limited applications due to severe nephrotoxicity, cardiotoxicity, and the toxicity nervous system and bone marrow. Now, Fundisom was Indian drug. It's a trade name of Indian drug, which was developed by uh, Professor B.K. Bachawat, Department of Biochemistry, Delhi University. It was a cost-effective substitute of Ambisom, which is US competing product. Indian government's Department of Biotechnology supported this research, and then clinical evaluation of this product was done in KEM Hospital, Mumbai, and then different hospitals of Mumbai. And clinical trials for Kala Azar was led by other team, which was comprising of Dr. C.B. Thakur, who was former Health Minister of Government of India. After completion of clinical trials, it was given to NRDC for technology transfer. Then first transfer happened to ACE Diagnostics. By the way, it couldn't succeed because of various market reasons. The second transfer happened to Life Care Innovations in 1999, and then they improved this formulation further by overcoming doses which can affect toxicities. And then support from Government of India Department DSIR was taken from a program called PETSER for this research work. This product could be having safer toxicity levels, and it could be having lesser cost, less than 1/10 of the cost of the imported drug. And then this could be used for treatment of diseases like systematic mycosis, Kala Azar, and burn injuries. And the next technology is UVC-based disinfection trolley for hospitals. This is developed by my lab, ARCI, University of Hyderabad, and a company called Makins Industries. What is happening actually? Our beds in hospitals are limited, and then they need to be, because present COVID situation requires the beds have to be vacated, or maybe kind of cleaning also has to be done very fast. Then this UVC based trolley can disinfect this kind of beds and rooms very fast. And then the UVC based trolley having dimensions of 1.6 meter, width of 0.6 meter, and then length of 0.9 meter, they can disinfect walls as well as the lower side, the floor also. And then field trial in this case was done in a hospital called ESIC hospital in Hyderabad. And then if you just see the field trial results, this can disinfect more than 99% in 30 minutes a room of 400 square feet, and when this trolley will move at 5 feet per minute. Now, concluding thoughts, there is a need to enhance the trust between different stakeholders, including academia, industry, and R&D. We have to identify the crucial links which will connect innovation chain from ideation to the technology transfer, because no chain is stronger than its weakest link, as all of us know. Then we have to identify a business model. How do we leverage the capability available at different levels of R&D? And then what kind of partnership will help to leverage this research capability? And then for this, we need to experiment. We do testing with the different business models, different partnership situations. In my view, the lab test with the material and the test with this kind of business models are to be given, both are to be given due importance. Then only we can have impact of the research capability available in the labs, whether it's in-house R&D lab or maybe external laboratory. We should continuously evolve formats to address emerging partnership situations. And then we should have flexibility in forward progression, because based on the outcome at different stages, different levels, we should have a flexible strategy. How do we take forward to the next level so that we can have more and more impact of the research in the lab? And last but not least, it's very important. There are the opportunities for pharma professionals to add value in the chain, which is starting from ideation which goes up to technology transfer and commercialization at different points, as I have mentioned in the chain. And then I think you can contribute in any place depending on the interest you have. And then in my view, if you can prove that you're adding value to the chain in some way, the compensation will automatically come to you. Compensation means monetary benefits or salaries and all that will automatically come to you if you can prove that I have made a constructive contribution to this chain, which is starting from ideation to technology transfer and commercialization. And then there are IP firms, there are labs like ARCI, who are employing people with different backgrounds, technical backgrounds, with an idea of how to commercialize them, how to transfer them to industry, how to make uh, derive more and more value, how to monetize this research. And then these three papers and then one short article we have written during last few years, which can help you to implement these thoughts in your respective organizations, whether it's research labs or companies, so that you can derive more and more benefits from the research capability available in your organizations. I acknowledge the kind of support given by director of ARCI for implementing this model during last 6 years, and my colleagues also who have cooperated with my thoughts, so that they could derive the actual value whatever we intended 6 years ago from the research they were doing. So thank you very much for your kind attention. And then, yeah, I am happy to take up questions. I think probably question answer session is being arranged in the end of my fellow I think speaker, Dr. Gaurav. After he will finish his talk, that time we will have Q&A session also. Thank you very much for your kind attention. It was a pleasure to interact with all of you.
Thank you very much, sir, for guiding us regarding technology transfer. The basics and the case studies were really useful to understand the basics of technology transfer, that how research can be go in the commercialization. Gaurav is also connected with us. So, sir, you just stayed in the background. So once the Gaurav sir session will complete it, then we'll go for the question and answer, sir.
Sure,
Okay?
sure.
Sure,
Thank
sir.
you.
Sure. So with this, we are moving ahead with our session. The next part of the session is going to taken by Dr. Gaurav sir. Hello, sir. Yeah, great. So before just transferring this control to Dr. Goel, I just want to introduce him that Dr. Goel had completed his doctorate and master in pharmaceutics from ICT Mumbai. He's also completed his bachelor's from Jamia Hamdar University. Both are, we can say, reputed institutes of our country. He has a total 13 years of experience in industrial and academic research areas. He's worked with renowned industries just like Akums Drugs, Rusan Pharma, and he's also currently working as head of technology transfer and department at Silpa Medicare, Bangalore. His focused research areas are development of oral disintegrating films, transdermal therapeutic systems, implants, nasal sprays, lyophilized injectables, etc. Dr. Goyal is the first Indian scientist who launched indigenously developed generic fentanyl transdermal patches back in 2009 in association with Rusan Pharma. And his topic of discussion is a checklist for a successful technology transfer. So Dr. Gaurav will guide us ahead with his session. Sir, up to you, sir.
Yeah. Thank you very much for the introduction. I'm very thankful to Dr. Sanjay Bhardwaj also from ARCI, uh, who has wonderfully and very lucidly explained how technology transfer, your technologies can be commercialized. Because in India, lot there are many minds who wants to who has the technology with them and they want to commercialize, and they really need to understand how technology can be commercialized. And your lecture, I definitely can say it is very wonderful and very useful, knowledgeable for them to understand how there are what are the different government agencies who can support for the commercialization of the technology transfer. My presentation is basically a checklist for how, before we do some technology transfer from one side to the another side, or from the R&D or academic level to the institutional or industrial level, what are the points which we should consider before going with the technology transfer? And the word which is catchy here is the successful, because we need to do the successful transfer, because many people, many academicians go for the technology transfer in industries and they generally come back with the failures that this technology is not working in the industry. So before that, we should understand how we can make this technology transfer successful. What are the various small steps which we need to think of to go with the technology transfer from one side, like the site which has the technology is known as the supplying unit or sending unit, SU, and one where the technology is going to be transferred, it is known as the receiving unit. So these are the two terminologies which is internationally applicable, SU and RU. So with this, I will start with my presentation, and I really want that audience should ask the questions relevant to the if they have faced any difficulties in the technology transfer and they want to understand that, because I have taken this particular subject because during my PhD I did a first technology transfer of indigenously developed transdermal patch. So itself I have not seen the transdermal patch, and to understand and to do the technology transfer, I made a contacts with some US friends, and then I took the patches from them. So these are the for a successful technology transfer from the academic level to the industrial level, you really require uh, uh, you can say Junoon should be there, so that it should be able to do the successful technology transfer. So now with these words, I start with my presentation, and I want it should be in the discussion mode later on, right? So if you see what is technology transfer, it is a logical procedure that controls the transfer of an established process which is developed into the R&D through documentation and it is required a professional expertise to the site which is capable of reproducing the process and its support functions to a predetermined level of performance. Like whatever specifications, whatever the level you have decided that the product should be up to that particular performance, that company should also have, the receiving unit should also having the capability of reproducing that particular process. What do you mean by successful? Successful means when we say our technology is successful when it is validated. When the process gets validated and you are getting the reproducible results into the receiving unit, then we can say it is a validated or successful transfer. So it should be documented through the process validation protocols. Now we see pharmaceutical development takes place into the uh, academics or in industries or on one side on the drug substance or either on the formulation. We do some clinical supplies, small drug delivery systems or analytical method. Also, we develop some analytical method. Some industries require the analytical method and they do not have the capability of making the analytical methods. So technology transfer can be done on the analytical methods also. Some packaging support, some companies required from outside agencies, so that can be taken. So these are the technologies which can be developed at the industrial level or at the academic level, which can be transferred into the industry level. Then the next phase is to the verify, that is the technology transfer phase, where we do the performance qualification, which is also known as process validation of that particular product or technology, where we scale up that technology into 10x or 50x from the very lab level to the commercial level, or maybe the pilot scale level into the industry, and we do the performance qualification, which is also through the process validation protocol. And this particular phase is known as basically technology transfer. And if it is this particular phase, the product is qualified and the process is qualified, then we start with the commercial manufacturing, and then it goes into the manufacturing. So why technology transfer department is required? Initially, all the R&Ds used to do their own technology transfer into the industrial scale up to the commercial scale. But now a very specific team, which is known as technology transfer team, is required for the technology to be transferred from the R&D level to the industry level. There are various reasons, because if the technology is not is having the proper knowledge, and the person who is not having the proper knowledge of doing the technology transfer, such what type of problems which we can face is we can have the sub-robust processes where the performance ability will be decreased. We have the reduced production rates into the industry, the production which is increase the cost of the production, increase the number of atypical performance into the product, like product defects, elegance issues, decreased process reliability, not being capable of handling variations of raw materials, API, process controls, operators, etc., inefficient validation, and many more. So if you will see this, if we have the poor technology, or if it is not transferred properly, if we do not have the control on our technology, then such type of problems can be faced. And so to avoid such type of problem, how we need to what are the checkpoints for the successful technology transfer? This which we are going to start with this. So what is the importance of successful transfer? That we should give the product with the quality, with the high speed, and the result should be into our predetermined level, that is our predefined specifications, and the cost should be reduced. Cost automatically reduced when we give the quality product at a speedy rate. So if your technology is successful, and definitely the cost will decreased on the longer term. So here is this Ishikawa diagram, which is known as fishbone diagram also, which is generally these are the 6 factors which are responsible, which are the major cause for building the quality into the product. So this is as per the ICH guidelines. So these are the factors like which we need to consider to make a quality product. These 6 factors are man, material, environment, measurement, method, and machine. So we go with one by one and discuss on these 6 aspects, how we can, what really needs to be seen before doing the technology transfer, and go for the technology transfer. So first and foremost is the agreement. There should be an agreement between the supplying unit and the receiving unit, which is also been known as the confidentiality agreement, technical agreement, mutual understanding between the two organizations. And this is basically agreement for controlling or transfer of the intellectual property from supplying unit to the receiving unit. And this is based on some royalties, and there are various modes which Dr. Bhardwaj has discussed with us. And the licensee attains the receiving unit, that is the licensee attains the right of use that particular technology for a fixed period by paying a particular fee for it. So this type of agreements should be in place, so that after that technology transfer, some conflict should not be raised. Now the first factor which we need to discuss is demand, which is very, very important aspect of having a successful technology transfer. Whenever we do the technology transfer, the first factor which we need to consider is the manpower training. The manpower to be trained of the receiving unit from the supplying unit. This how supplying unit need to train the manpower either through the PowerPoint presentation made by the supplying unit to the receiving unit on the product, on the process, and that is very important. The more and if it is feasible that if process can be demonstrated at supplying unit to the receiving unit persons can go from receiving unit or through video also they can demonstrate the process at the supplying unit. So at the lab scale, if they can show that demonstrate the process, it will be easy for the receiving unit to adopt a particular technology, or the transfer protocol should be in place, which is a written documentation. Basic aim of technology transfer, it should be documented properly. All the things, what are the stages and how the particular technology transfer will be taken place at what will be the different phases, that all needs to be covered into this transfer protocol. The very important thing is which is required is the qualified and the experienced manpower at the receiving unit is very much important, because the technology which is supplied from the supplying unit, the person at the receiving bay should also be should able to understand that particular technology and the importance of that particular technology. So it should, a qualified and experienced manpower, if it is there, it is always easy for the supplying unit to transfer the technology. Then documentation team is very much essential, because whatever technology which we are doing it should be all the things, all the observations should be documented properly. In the pharma industry, there is very good setup for its documentation team, and then it generally does not creates any problem, because it's a very QS is always there for the documentation. Then training of documentation by the supplying unit is very much essential, because this is very important point, because we generally see generally taken granted that receiving unit will document all these things, but what things to be documented, what observations needs to be documented, and how it needs to be documented, what calculations, if there is a API calculations also. There are various ways of doing the API calculation when we doing the dispensing, before dispensing of the API for the batch. There are various on the LOD basis, on the moisture content basis. So there are various type of calculations which are there. So the training from is very much required by the supplying unit to the receiving unit for proper documentation and filing of the records. Next is the materials, which is what we need to do. What is the checklist for the materials? So we need to understand first the design of the specifications, right? So the person who is responsible for technology transfer, he should understand, because the for all the raw materials, the vendor already has their own specifications, or the specifications are well mentioned into the pharmacopoeia monographs. But what specification is very much critical for controlling the quality of the product? That is very much essential, because one specification can be important for one of the formulations, but might not be very important for the other type of formulations. For example, if we say particle size distribution of diluent like mannitol, it is not playing a very significant role during the manufacturing of the conventional release tablets. But if we see this particular particle size distribution of mannitol for a perspective of oral disintegration or mouth dissolving tablet, then the particle size for the mannitol plays a very important role. So for one of the formulation, particle size of the mannitol does not play any role, or does not having an important role. But for oral disintegration tablets per se, it's playing a very important role. So the technology transfer person is very important, plays a very important role over here to understand which specifications are very much important for a material to keep in so that the technology can be transferred successfully. The similar example is BET test is not required for the excipients, which is for the aseptic or sterile manufacturing, where it is very much required for the other dosage for the sterile dosage forms. So it is the BET test should not be there if it is for the other dosage forms. It only should be there for the sterile or aseptic manufacturing products, right? So I have seen some specifications recently. I am putting this particular example because I have seen some excipients which is having the BET test, and we are making for the oral use products. So this test is by the default the QC chemist has added, and I told him to remove it because then BET test does not play a significant role for the oral solid dosage forms. So such type of things needs to be think of before deciding of the specification. One more thing, the specification should be consistent with the reference batches or developed batches, bio batches, which we have produced at the supplying unit. Because this is very important, the material which we have taken, the vendor which we have selected for the during the development, it should be the specification of those materials should be exactly same. If you are changing the vendor also, you try to match those specifications so that you can get the consistent product. If that particular specifications plays a important role, like I have just explained it above, like this is a very important critical the specification which plays a critical role in that particular formulation, those specification needs to be matched, right? And one more thing which is very important with the percentage of the material, because once you develop the technology and you do not know what is the regulatory compliance for that particular, how much up to what percentage that material can be used, or up to what milligram, or what up to what level it can be used. So this is one of the example how we can see there is one US FDA website which is known as the inactive ingredient search, IIG limits, which are there. And if you see, there's a on the screen there's a copovidone which is used in capsule for 224.4 milligram per dose. That means up to this level of copovidone can be used into the capsule formulations easily up to this particular level. If it is not there into this particular list, then you need to go through the safety and toxicity studies, right? So then you have to prove that, and then you have to generate the data on safety and efficacy studies. The next important thing which is very critical for a successful transfer is the environment. Environment, if it plays a very important role, like what is the pressure differential required from the corridor to the room, manufacturing room, because environment is very not only important for the product, it is for the safety of the manpower who is manufacturing into that particular manufacturing unit, and it is also controlling the cross contamination between the different products. So you should know how what environment you require into your facility, and what differential pressure is required for manufacturing of this particular dosage form. Temperature, humidity, one is the vacuum range. What is the environment for vacuum range if you want to deaerate something, or if you want to transfer from through vacuum line? What is the vacuum requirement? Because the vacuum requirement is very different for powder to powder. Some powders are very bulky, some powders are not bulky, they are heavier. So some we need to understand that what vacuum is required for transfer of our powder from one side. Because vacuum range is also very important, because if you apply a higher vacuum, there are chances of segregation of your blend. So, this is also plays a very important role. What is the vacuum requirement into your line, right? If the product is sensitive for the oxidation, then do you require nitrogen or any other special facility that is very important. The nitrogen, we should understand the percentage purity of the nitrogen should be high. If it is high, very much sensitive, because like for the sterile dosage forms, the nitrogen requirement is 99.99%. So such type of requirement should be seen before doing the technology transfer. Then what is the area which is required for the manufacturing of that particular formulation? Flow of materials, how the material is going to flow from one area to the another area, and the major role is during dispensing is of the environment, because during dispensing the laminar airflow is required, or not, because it is also very important for the safety of the manpower. Next comes to the measurement, uh, which is plays a very important role for a very successful technology transfer. Like what parameters you are controlling in process during the manufacturing of a product, because later on after the product is manufactured, then it is go for the finished product analysis. But during the manufacturing, what parameters which we can analyze in between, so that we can assure that the manufacturing is going properly. So the major thing which we can see, if for example I have just highlighted here with the example of a tablet, like if we measure the four major things in process, like bulk density, tap density, and moisture content, or either LOD of the bulk, or particle size distribution of blend, then by these four parameters we can very much assure that our finished tablet formulation will be having a very, we can control our finished tablet formulation with the quality, right? uh, because then the process of compression is there, which we can control through there are pressure gauges and all. But if these four parameters are not controlled, then if it is out of the specifications, then definitely it will impact on the quality of the finished product. So for a tablet formulation, generally these four parameters are important. So in the similar way, if we say liquid, the viscosity and pH of the liquid controls the quality of the liquid dosage form, and as well as it will control the stability of the pH, because the pH is very important, plays a very critical role of the stability of the active into the liquid dosage form. If we if we having the control on the viscosity and the pH of the formulation, then basically we can guarantee up to the more than 80% that our this liquid dosage form is almost under the control. So in the similar fashion, that we have to understand what in process specifications are required to be measured, so that we can assure our finished formulations. Now, nowadays, companies or industries are starting up with the PAT technologies, process analytical tools, where is a near infrared spectrometer is the very commonly they are using. It is the inbuilt into the machine, and they are observing whether the bland uniformity is attained or not. Like there is an example which is there into the chemistry lab example, where they have used in NIR this, which we can see this the large diagram, the extracting unit. There is a line which is going through the PTFE tube inside the uh, through the UV spectrophotometer, and then through the NIR, to understand whether the reaction is completed or not. So this it is the it because before that it is you have to take out the samples and then run through the uh, your chromatography. You need to do the chromatography and try to understand that whether the reaction is completed or not. But during the reaction itself, you can monitor whether the reaction is completed or not, and whenever the reaction is completed, then you can stop. So this is a very it saves the time very much, and with a very more accuracy. So such type of equipments, or if it is there in the industry, or it is a technology transfer team, the responsibility comes under the technology transfer, that such type of proposal should go to the management to have such type of things where we can improve the quality of the product. A similar example which is shown here is the band uniformity, where there is a NIR spectrometer on the blender, which is installed in the blender, V-shaped blender, and we can understand when it is in the uniformity, it will show the uniform results. So through the NIR spectra, we can understand these things. Next is the method, where it is a very important role. Method is the one thing which is responsible, which is actually, there are two things. One is the method, analytical method, and the process. But that is the manufacturing process. These two things needs to be transferred from the supplying unit to the receiving unit. So what is important in that, and what needs to be checked before transferring the method? That process or method should preferably be developed as per the receiving unit machine suitability. This is very important. If you know your client, if you know where you are going to send this particular transfer the technology to, what will be, who will be your receiving unit, then it will be very easy for you to so that a supplying unit, that is the academic institution or either any other CRO who is developing the technology. Yeah. Uh, so here, what we need to see this method as per the receiving unit machine suitability. A design space to be properly defined. What is design space? We all know, it is very clearly mentioned in the ICH guidelines. The design space is the space where if we keep our process into those at particular, between that particular range which is defined as a design space, then we can get our process performance and stability of the product. A design space should be properly defined by the supplying unit, up to what range there you can do the homogenization, what will be the time, what will be the temperature. Design space of all those critical process parameters should be properly defined, and and value of any process should be always defined. So if any technology is to be transferred from supplying unit to the receiving unit, we should know what is the and value of that particular process. When we say this process is completed, like for example, if you are doing some drying step, you should know what is the moisture content or LOD. It should be properly defined through whatever trials you have taken at the supplying unit. You should monitor those moisture content and LOD, and it should be controlled. And then design space should be defined properly. Up to what level it should this particular LOD can be kept into the finished formulation. The design space, it should not be like that. You give. I have seen some companies are making some specification that not more than 10%. Not more than 10% means it can be 1%, or also it can be 9% also. So if it is working at that particular range, we should have some results of giving that particular design space that not more than 10%. Generally, a range should be given that to what level the moisture content should be permissible. For homogenization, like if certain people are doing homogenization and they are just mentioning the time that do homogenize for at 3000 RPM for 20 minutes. But in the industry, when the homogenizer is of the different make is there and different principle is there, and 3000 RPM for 20 minutes may not give you the same sort of results. So therefore, we need, when we are developing some technology at the sending unit or supplying unit, we should have the particle size distribution and to be measured. What after homogenization, what is the particle size range we are getting? What is the distribution of the particles? so that we can say when we are going to transfer the technology to the receiving unit, we should have the same sort of particle size distribution, similar type of particle size distribution. Or if particle size distribution is not facility available at the sending unit, then you should uh, measure some equivalency, something like that, measurable parameter like viscosity or sedimentation rate, where we can say which is a direct parameter which is showing, which is proportional to the particle size distribution. So if the particle size distribution, if it is not there, we can then have some other parameters like viscosity or sedimentation rate, where we can say that our homogenization process is completed. Method should be such that the RSD between the measurement should be minimum. When we are when we are doing some reproducibility experiments, we should see that we should always calculate our RSD of the experiments of that particular step, and when it should be seen that the RSD should be minimum. For example, drying at 60 degrees Celsius for 30 minutes can be more efficient to produce lower RSD in comparison to drying at 70 degrees Celsius for 10 minutes. Like if we are doing 60 degree heating drying at for 30 minutes, RSD we are getting suppose 3%, but 70 degree Celsius for 10 minutes we are getting the mean value around same, that is the moisture content is 5% in both the cases, mean value is same, but the RSD in the 70% maybe in comparison to 3%, it must be 5% or 10%. So there can be, so we have to evaluate, we have to make a process such that we have to select a process where the RSD is less, so that we can get the reproducible results. So these add values are very helpful in optimizing the process parameters during the scale up. Verification of design space should be done during scale up or trials at receiving unit also, because design space is given by the supplying unit must not be there as a design space for the receiving unit. So at the receiving unit also always the verification of design space should be there. Some methods are adopted to get more uniform blends such as geometrical or sandwich mixing. So we have to adopt some methods like which during the, if it is because the supplying unit level is very less, or it is at X level, but when we are going for the scale up at 10x level or 20x level or maybe more than that. So generally we can see we have to think of some methods where geometrical mixing or sandwich mixing may be required. So we have to think from that particular angle. So this comes with the experience. In case of complexation such as saccharoduction or ion exchange resin complex, when we are making, we have to optimize certain parameters like optimization of the time, volume of diluent, stirring speed and pH, which are very, very important parameters to get the reproducible results from sending unit to the receiving unit. Otherwise, we can get variable results of dissolution or either in taste after a transfer also in the commercial batches. So this side, then again it goes back to the R&D. So to avoid such type of things, we need to optimize all those things. These parameters like stirring speed, pH, which is very which seems to be a simple parameter, but plays a very important role in getting the uniform. So with such type of technology, first of all, we need to understand our method. For that particular method, what will be, when we can say this method, what are the influential parameters? So these are the different parameters which influence the saccharoduction complexation and the addiction resin complex. So with this type of example, I want to say that we have to understand with like for other things also. A discriminatory ratio of dissolution media to be proved so that the poor formulation can be identified. We have to select. This is a case where I'm seeing many of the companies, pharmaceutical companies or academic institutions, they do not understand this. They generally select the media which is giving the very fast release of, but you want that particular fast dissolution media will not be able to discriminate the poor formulations. So we have to select a media which is having the discriminated nature. One of the very important thing is the cleaning method. The cleaning method should be properly developed and validated before transfer of a batch from supplying unit to the receiving unit. Because if we do not have a very proper cleaning method, and if the supplying unit is not giving the cleaning method to the receiving unit, it can lead to the cross contamination of active from this particular formulation into the another formulation. Not only active, it can be inactive also. Yeah. So analytical method should be validated before taking the exhibited batches. This is one of the very important checklists for scale up or taking the exhibit batches. Then we come down to the machines, which where the machines basically, if we say the most favorable case is the sending unit, the receiving unit should be the equivalent. The machine facilities or the equipment should be equivalent. This is the most preferable case if the industry is having their own R&D center, and then this type of thing is definitely can be feasible. Batch size selection is mainly dependent on machine capacity or process reproducibility. Uh, and batch size should be such that the proper process can be run. For example, if batch size is too high, equal to the working capacity of the container, there are chances to get uniform blend in higher time than 60 to 80% of the working capacity. What I mean to say that batch size is very important for getting the reproducible results, or defining the lead time of the particular process. Because if batch size is very high, the time to get the uniformity in the blend is very high. Whereas in case of 60 to 80%, the blend uniformity can be achieved in the lower time. So this comes with the experience, how the product is performing. Very important in that. A gap should be analyzed before transfer of supplying technology from the supplying unit to the receiving unit. What are the very critical elements, what are the facilities available at the receiving unit, and how this technology is developed at this supplying unit. So those gaps should be properly documented, and technology transfer team should work on that particular gaps so that the technology can be properly or successfully transferred. So if I want to summarize these particular things like what we have discussed during the bulk manufacturing, the product and master formula should be there. This is the checklist for the technology transfer. Manufacturing work instruction should be there. Cleaning method should be in place. Machine qualification should be completed. Packaging work instruction should be there. Dedicated process equipment, if it is needed, then we have to mention to our receiving unit that this particular units will be required for dedication, otherwise contamination can occur. And MSDS, how to handle that particular product at the uh, production facility, that is very important, because it is related to the safety of the person who is working with that, and the forecast and volume should be there so that we can understand what batch size needs to be kept uh, for the batches. In case of packaging, if we say it is, we need to have this source or approved vendor suppliers names and address. The qualification, vendor qualification should be in place. Specification and certificate of analysis should be in place. Test method and reference standard should be in place. MSDS of the material should be in place if it is hazardous material. Primary packaging components and specification should be in place, including the packaging insert. And secondary component specification should be there in place. Largely specifications of our finished product, bulk product specification should be there. Finished product, that is the release specification, should be there. Stability specification should be there. Cleaning validation documentation should be properly documented. Packaging validation documentation should be there. Process validation documentation should be there, and unique handling needs, that is MSDA, should be documented properly. So what is the intake for the academicians? This is all about the technology transfer checklist. Uh, this is the last slide of the presentation where I want to say for the academicians, what is the, with my presentation, what is the intake for the academicians? That first of all, academicians should understand your expertise, because all academicians who are working in colleges, they always have the dream that they can able to see one or at least one of their formulations working in industry, because they are doing very good research work, they are making good publications, but the end result is they also want that they one of the formulations they should develop whatever they have taught to their students, they can see that particular practically on floor. So what I want to say to the academicians by this particular by this my presentation, that you understand your expertise first. Don't see from to which industry you want to do the technology transfer. Don't see from particular formulation. First of all, understand what is your expertise. If your expertise is lyophilization, is your expertise is complexation, if your expertise is ion exchange resin complexation, is your expertise in making microreaders, what transdermal patches, what is your expertise? Like then understanding your expertise, then see from another angle. Like for example, I have given in the second line, like you can, if you are expertise, if you want to make some liquid formulation also, you can make a very simple paracetamol pediatric suspension also, which is required by most of the companies at present also. You believe me, paracetamol pediatric suspension is not easy to make. It is having the stability problems, it is having some sedimentation problem. So if you can make in collaboration with the company who is having the liquid facility but does not have this particular formulation, so you can ask to them, you can contact them and tell them, I have developed the paracetamol pediatric suspension, and or either the way you try to talk with them, you have the liquid facility, which type of formulations you want, which type of formulation you want, because most of the countries still, if you see 80% or more than 80% of the Indian companies which are very small and situated in the small towns or cities of Ludhiana, Chandigarh, near Delhi, Noida, to and in Maharashtra, there are many companies, Gujarat, which do not have the R&D center of their own. So they, if you can just talk to them and just collaborate with them, what formulation you require, we develop this for you. Just give us the API, because you are already making some tablets of paracetamol. Give us paracetamol API, and we are going to develop it for you. So such type of understanding definitely can help you out to do some technology transfer. Uh, Similar examples, the metformin, the glimepiride tablets. Still, if you see, there are many brands which are available for metformin and glimepiride tablets, but glimepiride belongs to the BCS class 2 molecule, and it is having a very poor solubility, and uh, and that is why this is one of the reasons this metformin and glimepiride tablets is not effective of all the brands. It is very much the only few brands which are very much effective for the glimepiride tablets. We have to understand that this type of formulations, if we can develop also, or either there are some bilayer formulations, and many companies does not have the bilayer manufacturing facility, and if you can convert it to the single layer tablets and showing the stability, that can be added advantage to their particular company, and that they can launch their products. So such type of things, cosmetics and herbals and nutraceuticals is one of the sectors where there are less of the regulations at present in India, and you can easy to launch those products. Similarly, and many more things which you can make it to try to understand your expertise, and develop the product accordingly, and then do the technology transfer. So small technology transfer can give you the confidence, right? For example, I just want to say, like just a year before, I was working with an college, ISF college, Moga, Punjab. Right. So from there, I did a 3 technology transfer within a span of 9 months. So if you have the collaborations and if you can do develop the product, if you have these stability chambers and all, you can definitely can do from academic institution and can do the technology transfer. So with this, I want to thanks to the audience to listen me, and thank you very much.
Thank you, sir, for this very informative session. I think all the academicians who are there, and this session will also be live. This session will be also recorded at YouTube at the end. So definitely everyone, not today, but after the time period also, they will get this important information from this session, and they will come to know that how this industry is actually working and how they can contribute even by staying in the institute level, which is a very common in the US countries like US or UK. This practice is very common over there. But in India also, since patent and these things came in the picture since last 4 5 years, UBC and AICT is also working on it. So definitely, institute level academicians are also working on it. So definitely, we'll go for the quick question and answers. There are a few questions came from audience. Some are there from our side also. So first, we want to ask Dr. Sanjay sir.
Yeah.
This question came from Vandana Jalod ma'am from Vital Pharma. That are you providing third-party transfer and third-party consultant expertise for global regulatory?
Yeah, that actually we will see the government regulations, because we have not done actually much work for outside India. Actually, most of the companies for whom we have worked there in India, even for example, like companies like Boeing or GE, they have office in India. Those people we have done, but somebody just exclusively having office in any foreign country, we have not done, because we have some actually kind of governing rules from government of India. Under the rules, we can always explore the possibilities, certainly.
Definitely. Thank you for the answer, sir. Then the question is for the Gaurav, sir. That because so many students are attending this session, we just wants to take a highlight on career aspect of technology transfer in pharma, sir.
This question is very relevant, seeing the current scenario. Technology transfer, now companies before, if you see a decade before, very few companies is having the technology transfer department. Generally, the R&D, generally people do the technology transfer in the industry directly. But now, this technology transfer, all the major companies who really wants to go for the regulated markets, wants to file for their products into the regulated market, they have a very well set up for the technology transfer, so that the a technology transfer person understand how the technology needs to be controlled, and it's regularly monitor the product also after the post commercialization also. So just a minute. Presentation presentation also, right? To discuss the yeah.
Yes, sir. Please continue, sir.
Yeah. So I want to say, yeah, sorry for the disturbance. Like, actually, I am working in a company.
Yeah, we get that, sir.
So I have not took the leave today just because of some important meeting. But definitely, this technology transfer, as far as the career perspective, I can say it is having the good perspective. But first of all, you should having the good experience of supplying unit. That is the R&D center. Then only, like after spending 15 years into the R&D, then I switched over to the technology, because you should understand the both the things. the receiving unit requirements also, the sending unit requirements also. So directly coming for the technology transfer is not the ideal into the group. It is generally an experienced professional is required.
And sir, which qualification is best if someone is going for masters after pharmacy? So which is the best suitable for this particular field?
Definitely for the technology transfer, when we say it is a technology transfer, then there are two technology which needs to be transferred. Basically, it is the process and the method, analytical method. So if you have an expertise in analytical method, then also you are suitable for this particular technology transfer process. As well as if you are good in if you're from the pharmaceutics background, then also it is very beneficial for you because you understand the machines, you understand the processes.
Okay. The next question is for Dr. Sanjay sir. That what are the tools used to know readiness of R&D? Means how we can just understand at the one level that our research is ready to go for the commercial. What are the tools? How we can map that?
Yeah, so we can have assessment of R&D in the lab, and then what we should have continue. We should have some kind of perform or template for our research teams. When then this will help us to know. And then so many questions. By the way, you ask your research team, you will know at what level is this research. For example, somebody says my research is at level 6 in my levels. Level 6 means somebody has to do some field trials, and then we may ask the team, yeah, what kind of field trials you have done? What are the results of those field trials? In case of pharma, it's clinical trials, drug manufacturing. Then if team is saying, yeah, this is a clinical trial we have done, what kind of feedback you got from those companies or maybe users? And then this will help you to assess whether they have successfully passed level 6. And then when you go to level 7, for example, then you have to repeat those clinical trials. Like now we have phase 1, phase 2, phase 3. And then based on this kind of outcomes or maybe feedback from the user entities, you will be able to determine what level your R&D is. And then after your R&D is over, your field trials are over, then you can make a business case, and that will be level 8. Then you have to have very close interaction, maybe a thorough interaction with the research team to determine the level of R&D your lab has arrived at in a particular research program.
Okay.
And then you can have some supporting mechanisms, whatever I discussed in my slides. Then you can have a support systems or maybe complementary capabilities you can find from either from your own organization or from elsewhere, then so that your team can move forward further in the research levels.
Okay. Okay, sir. Thank you very much for that information. Next question is for Dr. Gaurav, sir. That as you discuss about environment that can actually lead the product into quality. So same way can method is one of the factor that lead to quality of the product. This question came from our audience. Jitendra Singh has asked it. That method can also be important. That which method we should choose to get a quality of product.
Yeah, definitely. Method is one of the very important factor. Which method, like as I have explained you in my presentation, that quality has to be developed into the product. Which methods plays a very important role. The like controlling the method. Which method? First of all, like if there is a active, if it is a very potent drug and the concentration of the active is very less, then we may need to go with the geometrical mixing sort of thing or sandwiching type of thing. So the method plays a very important role. Like in fact, I can say one of the examples when I was working in industry and they are getting some hardness problem. I have seen that the method of cooking of the gelatin, because the gelatin was the blend binder over there, and the method of cooking of the gelatin was not proper. They are not cooking the gelatin properly. So this, all those things should be properly controlled, because one should understand what is the end of that particular completion of that particular method. Because once you know what is the end point of that particular method, then only you can able to transfer that particular method properly from one unit to the another unit. I hope this clarifies about it.
Yeah, exactly, sir. Exactly. There is also one question from audience that if this is for Sanjay sir, that how to consult industry for commercialization? Means
Yeah,
if you
there are.
have a product ready, how
Yeah.
they can contact? What is the standard procedure? How should they go?
Yeah. So if it is from a research lab, what they should do, they should identify what are the properties this product is imparting, what where it will benefit the most, and that's what we call the niche area, niche utility area. And then after you identify those segments, you contact with the properties of your product, that this product can help you to become more efficient, or maybe you can start a new product line based on this product. And then once you have, in my scale, if you've reached level 8, then you will have some kind of business case, also potential business case. Then if you combine both, your right match of the company which can use this capability plus a potential business case, if you approach these entities with those two or three things, then it will very high chances there that this company will be interested to commercialize your research. This is very precisely the approach someone can adopt. Again, this is from academia and R&D side. If it is from industry to industry, then again a similar approach I think should be useful, because you should have both. Your technical aspect should be satisfied, plus your business aspect. If you can qualify on these two main parameters, then I think they have very high chances that your any entity will be, or in terms of Dr. Gaurav's terminology, that receiving unit, any receiving unit will find it very attractive proposition to commercialize.
Okay. Okay, sir, that is great. And we will go for just a couple of questions. That one is for Gaurav, sir. That suppose, sir, I am a researcher and I am working on a bench scale activities. So how I can understand that how this product will be go at industrial scale, how it will perform it industrial scale or commercialized scale. How I can understand what is the gap in between these two scenarios?
Uh, this is a good question. Or every academician or every researcher generally have such type of question, because they generally we have the tendency, like every human being has the tendency, like they think that whatever they are doing is maybe not having the proper value, but they generally do not understand, or they do not know what value it keeps for the industry. Uh, so if you have, first of all, if you are developing some method in a college or at the academic institution, first of all, you need to understand some of the things which I have told in my checklist. The percentages of those ingredients should be as per the regulatory guidance, first of all. And foremost, which is very important for industry, is control of the related substances, that is the impurities. Impurity profile is very much important. If you are controlling the impurities into your process, then if you can prove that with this, at least industry does not require a very big stability data. For the initial discussions, they can go with the 1 month or 3 months accelerated stability data also. And if you can just produce 3 months stability data, first of all, you should know that your process is reproducible. It is giving the reproducible results. Once you understand and you have the confidence that this particular technology which you have developed, it is having a good, better taste, or is having the same taste as the product into the market, and or something better than that, or if it is not better also, if it is equivalent also, it is nothing, not a problem. It is generally industries require an equivalent product also. It should be look like same as the reference product. So if it is same as the reference product, so if it is reproducible also, 3 or 4 times you have repeated the experiment, maybe in your own laboratory you have scale up from 1x to 10x, then if you are getting the confidence, definitely you can start the discussions with the industry, with the small industries, because if your technology you think it is for the bigger industry like Dr. Reddy's, Sun Pharma and such type of technologies, they are like with the micrometers, patches and all, then definitely you can discuss with the big companies also. Like I did for the transdermal patch for fentanyl.
It's really great to know, and this is a very important aspect for those who are at a small scale level research or institute level research. They can exactly understand how they can go ahead with this. There is a last question for Sanjay sir, that how startup can do a transfer technology. Means if anyone coming up with a startup, Okay, so how they can approach you or any other technology transfer team, and how they can go ahead with their startup, how they can give funding and everything. Just
Yeah,
guide us on that,
now,
sir.
yeah. So now there's so many schemes available from Government of India also, which encourage the partnership between a startup company which is looking to commercialize research from public funded organization, including labs and academic institutions, and these startup companies. Then those schemes, I think I think that some company which is in startup category should first scan those schemes, and then they should then next is they should see the strength of the startup team. Because team capability is very important. Whatever is in line or consistent with this team's capability from a research lab, that will be having very high chances of success when they commercialize the research. So if then the other thing is having a complementary capability, because this research lab may have very good competence in technical terms or maybe research terms, developing new applications. But this startup team should have more of market orientation or application development, or maybe what are applications in demand. So then, if they have some complementary, as I have been mentioning, I think repeatedly in my presentation also, I think having a complementary skill and capability enhances the chances of a success of a startup. Then three, four things are, I think, the main ingredient for success of a startup. Whatever. So one is just to recapitulate. One is having a team which is highly motivated. Number two, scan for the scheme, scout for the schemes which are helping this kind of partnerships. And number three, having a complementary capability, whichever is not in the lab, and then your startup team is having, that will certainly enhance the success chances of the startup. This is what is a very brief response to this question from me.
Thank you very much, sir. Thank you. I just want to conclude our session with this. But before that, I just want to thank Sanjay sir. Thank you very much. I want to thank
Thank you.
Gaurav sir. Thank you, sir, for accepting our invitation at just a one request and just guiding us with this wonderful session. We learned so many things, and I think this will be just like a very great session for everyone who has attended, and those who will going to see it in the YouTube channel. It will be definitely be useful for them. So with this, I am just concluding session, and those who wants to have a certificate for this session, definitely they will receive it. We will send them emails on their mail ID, and they will just fill up the feedback form, and they will avail their certificates. Okay, so thank you very much, sir. Thank you for making this session and making this day wonderful in this COVID COVID-19 case. There, thank you very much, sir. I just want to
I'm
thank
Thanks,
you.
grateful to
Rajesh.
Dr. Rajesh and my fellow speakers. I think Dr. Gaurav, who complimented actually Dr. Gaurav, whatever I didn't cover, Dr. Gaurav complimented very nicely in his talk. It was a very nice match actually. I brought some other perspective, and Dr. Gaurav brought some other perspective to
Exactly,
this discussion today. And Dr.
Thank you.
Rajesh, I think moderated very well. And then thanks to audience, very nice audience we have today.
exactly, sir. Thank you very much, sir. Thank you.
Thank you, sir.
Have a
Thank you.
great day. Thank
Yeah,
you.
yeah, yeah. Have a nice day, all of you.